afternoon, everyone. Welcome back to many of you. For those of you joining us for the very first time, I'm Maggie Mahan, the Assistant Director for Community Engagement with the State Historical Society of Missouri. Thank you so much for joining us today. As the Associate Director of the Cape Girardeau Research Center, Bill Eddleman presents the 11th installment in his Beginning Genealogy Workshop series. Today's program will be an introduction to using DNA for genealogy. If you need to catch up on previous installments of this series or would like to rewatch any of the segments, you can view them on demand on the State Historical Society of Missouri's website at shsmo.org. All events in our virtual programming series are made possible thanks to the generous support of the Society's members and donors. Visit our website to learn more and see how you can add or renew your support. Thank you again for joining us. And now I will turn things over to Bill. Thank you, Bill.
Thank you, Maggie. Once again, this is the 11th in the series. There's only going to be one more in May. So I hope everybody has been able to either start at the beginning or go back and look at the recorded sessions so that you're up to speed. But today is we're going to talk about a topic that gets a lot of press in genealogy, and partly deservedly so, and I think partly it's a little overhyped, but it is useful, and that is genetic genealogy. So what we're going to talk about today and try to cover, first of all, what is DNA? Have to go back to some of that high school biology again. Can this tell me if I have Cherokee ancestry or Native American ancestry of any sort? Can it substitute for traditional research? Can I use it to construct my family tree? Do I have to exhume an ancestor if I want to determine if I descend from them? How private are the results? And this is something that's become more and more of concern to many people. What should I have done? There are a variety of tests that we'll talk about and the situations that you use them for. So first of all, go back to high school biology. And actually, if you're a little bit older than me, you may not quite have had this in high school biology, but what is DNA? DNA it's short for deoxyribonucleic acid. So that's why we use DNA. What DNA does is it codes for amino acids. Amino acids are the building blocks of proteins. So coding for a long string of amino acids ends up coding for a protein. Proteins are expressed as physical traits. And the easiest way to view this is something like hair color. Hair color has to do with the amount of melanin in the structure of the hair. And so protein equals melanin or the precursors for melanin. A lot of people use eye color, but it's actually more complicated with eye color than you might think. It's not just two traits, blue and brown. It's a variety of grades of that. It's actually under the control of different segments of DNA. But that's how we get our physical traits. It's all coded for in the DNA. That's what we need to make you, is your specific combination of what's in the DNA. In the cell, DNA is primarily in the nucleus, which is the brains of the cell, the control center. So that's one of the arrows is for the nucleus. There is, however, a small amount of DNA in an organelle that is a little subunit of the cell that has a specific function called a mitochondrion. For some reason, this still gets in the high school biology text and other biology texts that the mitochondrion is the powerhouse of the cell, without people knowing what the heck a powerhouse really is. But it's actually where energy is released so that the cell can function and so that you can function. So there's a little bit of DNA in the mitochondrion, and that's a special type of DNA that we'll talk about that's useful for certain genealogical applications. DNA is structured as a double helix. I always used to liken it when I taught introductory biology as a ladder that has to twist so that the rungs will actually meet correctly. So you have these two sides, which are structural in nature, and then in between those are the rungs. The rungs are comprised of bases, a type of chemical called a base. And there's 4 different versions of the bases, and we abbreviate those ATCG. But in order to get the structure to work, A has to pair with T, C has to pair with G. Otherwise, it won't work. And so the sequence of those has to do with coding for the amino acids. And because there's variation in the amino acids that can work in a given protein, you have differences in genetic traits that mark individuals such as you or me. The DNA is a long string, and it's packaged in a variety of different levels in the cell. First of all, a mappable unit of DNA. It's called a Centimorgon for applications for genetic use for genealogy. A Centimorgon is a mappable segment of DNA. Now, a lot of people get this, okay, it's a mapping segment, it must be something to do with distance. It doesn't really have anything to do with distance. It's just a mappable unit, a recognizable unit. It may be long distance, it may be short distance, but just think of it as a unit that doesn't really have a length component or a distance component. The number of shared centimorgons is a measure of how closely related two individuals are. Within that realm, DNA is packaged in units called chromosomes. And the number of chromosomes, we have 23 pairs of chromosomes in humans. The number of chromosomes has nothing to do with how complicated the organism is or how simple the organism is. It just is the way the DNA ended up being packaged. So as one example, there's an antelope species, which is a pretty complicated organism that has 2 pairs of chromosomes, 4 chromosomes. Well, it's just how it's packaged. It has nothing to do with its complexity. By the same token, there's another nematode, a little roundworm in the soil that has 220 pairs of chromosomes. Well, it's a pretty simple organism, but that's just how the DNA is packaged. We have 23 pairs. Now, there are a variety of different types of DNA that have to do with the function of the DNA and the location of the DNA that are useful to genealogy. And I'll talk about each of these, although I'm not going to talk much about xDNA. So we have Y-DNA, we have mitochondrial DNA, we have autosomal DNA, which is the big one. That's where most of the DNA is located, on autosomes. I'll talk about that in a minute. And then we have xDNA that's useful. So first of all, I'm going to talk about Y-DNA. Y-DNA in humans and mammals has to do with that 23rd pair of chromosomes, which determines sex. So if you have 2 X chromosomes in that position, you're a female. If you have an X and a Y, you're male. Now, admittedly, there are variations on that theme, but those are rare. So the Y chromosome, you inherit from your father, and if you're female, you inherited an X chromosome from your father. So in producing the sperm cells in males, you start out with these cells called spermatogonia, which are in the testes. And for whatever reason, a spermatogonium is stimulated to start developing. And it matures and becomes a spermatocyte. That divides, becomes a secondary spermatocyte. Those divide, and you end up with 4 eventually mature sperm cells. Because all the cell stuff outside the nucleus is divided evenly between those cells, those sperm cells, they don't survive very long. They're pretty short-lived for the most part. And so all of them are functional. Half of these end up being Ys. They get the Y chromosome because you have the chromosomes into these sperm cells. Half of them are Ys, half of them are X's. So when child is conceived, the male can contribute an X and a Y 50% of the time. Females, as we'll talk about later, only contribute X's. So when you put these together, you end up with 50% female offspring, 50% male offspring. The Y passes from male to son, to son, to son, and so forth. So the key point is Y DNA only works for male lineages. It only works from grandfather to son, to son, to son, to son, to son, and so forth. It may be necessary, particularly if you are a woman researching your ancestry and you want to test a hypothesis you have about a male ancestor, it may be necessary for you to find a male descendant of that ancestor. Well, it works pretty well if you have a brother. If you don't, you got to go farther afield to find another male descendant, like a cousin or a second cousin, or even more distant. And you have to convince them to test. So here's an example. And the hypothesis is that Daniel is the male ancestor of all of these people. So what can we use Y-DNA for? Well, okay, I want to, let's say my question is Sean a descendant of Daniel, and is Aiden also a descendant of Daniel? Can I test that with Y-DNA? No. Well, if we trace this down, Daniel has a son, Michael. Okay, so far we're good. Michael could test, and he's going to match with Y-DNA with Daniel. He has some sons, but unfortunately, Sean traces through Michael's daughter. So you break the chain. There's no Y-DNA. Sean got his Y-DNA from his father. Same thing if we go over here. Daniel has a daughter, Ellen. Well, the chain's broken there, so we can't use Y-DNA to test to see if Aiden is also a descendant of Daniel, as is Sean. We've got to use other methods, maybe non-DNA or other DNA methods. Okay, let's say we want to see if Brian is a descendant of Daniel and one of these other descendants of Bill. Well, in this case, Bill got the Y chromosome from Daniel. His son got the Y chromosome, and his grandsons also got the same Y chromosome. Well, if we go over to Michael and we're testing with Brian, Michael had a son who was the father of Brian, so the Y chromosome goes on through. Now, I will tell you with Y chromosomes, they do change slightly over generations, but for the most part, they're going to match up if it's from a common male ancestor. Common meaning the same male ancestor for different people. Also, you'll have some things called markers that will be tested if you do Y-DNA. The more markers tested, the stronger the case for common ancestry. Well, what is a marker? A marker is a gene or a DNA sequence that can be used to identify individuals. So it has to have, it's a sequence that, first of all, doesn't relate to male characteristics in general, because it's not going to vary that much. If you vary with one of those characteristics, you're not going to be male, or you're not going to express some of the characteristics of a male. It has to be something that varies, that's on the Y chromosome. There are other traits other than just male characteristics on the Y chromosome. So a marker is one that can vary without causing genetic damage, and but doesn't vary very quickly over the generations. So the more markers tested, the stronger the case. And I'll talk more about that in a minute. Also, for general ancestry, we can identify some things called haplotypes. These are very, at a coarse level, similar Y-DNA. So if we look at those at the coarsest level, worldwide, here's what it looks like. There are markers that identify the haplotype for those with ancestry in the Americas. So Native American, indigenous people to the Americas. Then if you go to the old world, there's an African haplotype, European, that is Western European, and Eastern European, and Northern Asia, and a Middle Eastern, an Asian, Indian subcontinent, East Indies, and Australian haplogroups. So this really doesn't tell you much in terms of your ancestry, unless you get somebody that you think came through European ancestry that has an African haplogroup or an Asian haplogroup. That tells you that somewhere back in the dim distant past, a male ancestor came from that area. So I'm going to work through some examples that I hope will make all this clearer for you. First of all, Thomas Jefferson's son was Sally Hemings. Then I have an example from my own ancestry with Eddleman's there are two different groups, one that's settled in Southern Illinois and one that's settled in Southeast Missouri eventually. So here's Thomas Jefferson's case, and this one was in the news and has been in the news quite a bit over the years. We know that Jefferson had no surviving male children with his wife. They did have a son who died in infancy, but his surviving children were both women, female. Two of Sally Hemings' sons, Eston and Madison, have male descendants that survive today in a straight male lineage. But as testing showed, there are problems with Madison. There was probably a non-paternity event, that's how they're called, at some point during that ancestry. So we're dealing with Eston's descendants primarily. You have some male descendants of Jefferson's father, though, through his brother. So his nephews, Carr and Woodson, and also of Jefferson's uncle, Field Jefferson, they're male descendants. There were also, the Carr and Woodson is one of the counter arguments as well. Sally Hemings' sons were from a relationship with Jefferson's nephews. And so they tested people who were descendants of Carrs and Woodsons as well. So the only match that was obtained was between Eston Hemings' descendants and Field Jefferson's male descendants. And at the time, that was 19 markers. That was pretty good for the time. Now they test a lot more markers. But even better, the paper documentation matches up. Jefferson kept detailed diaries. We know who visited at a given time at Monticello. We know when he was there. We know when he wasn't there. And as it so happens, when Eston and Madison were born, Jefferson was at Monticello 9 months earlier. His nephews were not there in every situation. Plus the Y-DNA didn't match. So Eston Hemings definitely was Thomas Jefferson's son using Y-DNA. The situation with my Eddlemans, I trace back to a Peter Eddleman who was in southeastern Pennsylvania and later Rowan County, North Carolina. There was another Peter Eddleman of the same generation who is pretty well documented as a separate individual who followed the same route. The only difference was mine went on to Lincoln County, North Carolina. Then a grandson of Peter ended up in Perry County, Missouri, and that's who I traced through. So Henry was my great-grandfather. The Rowan County man who stayed in Rowan County and died there had a son, John, who immigrated later to Southern Illinois, to Union County, Illinois. And the big question always was, are these two groups related? Or was it just happenstance that they happened to travel in the same area? Well, I tested at, what was it, 64 markers is what my maximum right now. And I have a friend whose mother was an Eddleman from the Southern Illinois group who convinced his first cousin, the son of his mother's brother, to test. We are a complete match at 37 markers. So it's the same family. The only thing is it goes back into Europe, Germany, prior to the 1730s, because that's when these men came from. Mine came in the 1750s. His came in the 1730s from Germany. Actually, Hessian part of Germany. So we were able to at least document that it was the same family. Some cautions about Y-DNA. You have several options for testing for DNA. Family tree DNA has been doing Y-DNA the longest. They have the largest testing database, long track record. Unfortunately, for autosomal DNA, they have a relatively small data set, but for Y-DNA, they're the best option. You can do a minimum of 37 markers nowadays, but test the largest number you can afford. And you can also do upgrades. They keep your DNA and you can do an upgrade later. If you have somewhere between 0 and 2 differences at 64 markers or 128, you're related. It's the same male descent. Surname projects are also an option. Some are more active than others. And it may require that you find distant relatives with male descendants to test, depending on what you're trying to prove. Okay, next is mitochondrial DNA. Mitochondrial DNA is in that little organelle within the cell called a mitochondrion, where energy is released. Biologically, it's thought that this is a remnant of potentially when mitochondria were a separate organism way back when, but they do have DNA. And let's go back to producing gametes, in this case, the female egg cell. It differs a little bit from the production of sperm cells. You have primary oocytes. A woman is born with all her primary oocytes, and certain one is stimulated to develop about once a month during her the period of time when she is fertile and capable of reproducing. And then it begins these 2 divisions to have the chromosome number. Only in this case, most of the cell stuff that it contains the mitochondria ends up going into one cell. Then with the second cell division, to fully have the chromosomes, all of it goes into one cell. So you end up with these 3 cells that are non-functional, and they're called polar bodies. So pretty much all the mitochondria are in this mature egg cell. So what does that mean? Effectively, you get all your mitochondria from your mother. Now, we found in recent years that there's some variation in that. Occasionally, there are mitochondria in sperm cells, but for the cases that we deal with in genealogy, that only throws a small wrench into things. It's predominantly from your mother. The key points here, it only works, in this case, for female lineages. And it may be necessary to find descendants of female ancestors through a straight female line. Well, as we know, if we've been doing paper trail, finding female ancestors is tougher than finding male ancestors as you move back in time. Females didn't leave nearly as many documents. The amount of DNA is far less, so they compare specific regions. But effectively nowadays, they test the whole mitochondrial DNA to make comparisons. The paper trail is still essential. Well, it's really essential with all genetic genealogy in order to clarify things. So here we go with another lineage. And if we look out on the right, the great-great-grandmother in red is passing all of her mitochondrial DNA to the great-grandmother, and then to the grandmother, to the mother, and then to the individual. Her brother gets all the mitochondrial DNA from mother, though. So I'm going to work through 3 examples in this case, all of which have been in the news at various times, that used mitochondrial DNA to test questions about ancestry. The first is Richard III. Richard III was briefly King of England. The War of the Roses, the final battle of the War of the Roses, took care of Richard III. He was killed at the battle. And a very nice Shakespeare play called Richard III. I guess one of the most famous lines is, a horse, my kingdom for a horse. Well, they kind of suspected where Richard III was buried, and it was under the floor of a church, which over time eventually evolved into a parking lot. And so in excavating this parking lot for a building project, they found a body which corresponded in the suspected location for Richard III's burial. And they were able to harvest mitochondrial DNA. In this case, Y-DNA wasn't going to work, but miraculously, there are female line descendants of Richard's sister surviving today. I believe there was one man who was a descendant and 1 or 2 women who were descendants. And the mitochondrial DNA in this skeleton matched with those descendants. And usually when you get a match with mitochondrial DNA, it's a pretty exact match because it doesn't change that much over time. Y-DNA changes a little bit more over time. The other one was the big question about Jesse James. Supposedly he was killed by Robert Ford, traditionally while hanging a picture up. He was shot in the back. There were various men that came forward in the 1950s, especially one old guy in Texas who claimed to be Jesse James, claimed to have bullet wounds that matched bullet, heel bullet shooting wounds that matched Jesse's, and claimed to be that, and his family always claimed he was Jesse. And so mitochondrial DNA testing came along, and somebody decided, we're going to test this. So they exhumed Jesse's body. So did he fake his death and escape, end up in Texas? He was in a wooden casket, and so when they exhumed the remains, they didn't find much except hair and teeth. Well, good thing. Teeth are very good about preserving DNA. So they tested mitochondrial DNA from the teeth and hair against mitochondrial DNA for the descendants of Jesse's sister, Susan. And they would have both gotten mitochondrial DNA from their mother, Zerelda, and she passed it on then to these descendants, both of whom were men, but they had female descent from Susan. And they matched to the mitochondrial DNA in the teeth and hair. That pretty much settles it in my book. But conspiracy theorists have come up with all sorts of harebrained reasons why, no, this can't be it. But I will tell you, they also tested the mitochondrial DNA of the descendants of the guy in Texas that claimed to be Jesse, and it didn't match. So it pretty much ruled out that guy in Texas as a Jesse James. Now, I'll say that I do have a connection through marriage to Jesse. A distant cousin of mine had a cousin, Reuben Samuel, who married Zerelda as her second husband. So that's my connection to Jesse, which is vague. The last one I'll talk about was Anna Anderson, who claimed to be the Grand Duchess Anastasia of Russia, of Imperial Russia, and that she'd survived the assassination by the Bolsheviks and ended up in Germany well after World War I in the 1920s, and claimed all of her life to be Anastasia. And some of the family members even said they thought she was Anastasia. Well, as it turns out, Anna Anderson had died before DNA testing came about, but they preserved, she had some intestinal surgery, and a tissue sample had been preserved from that surgery, where they were able to harvest DNA and mitochondrial DNA. Well, who do you test against? The Tsar's family is all dead, the immediate family, but she should have inherited mitochondrial DNA through the Tsarina, who is a matrilineal descendant of Queen Victoria. Well, golly, we've got plenty of descendants of Queen Victoria, and one of whom was a willing tester who contributed DNA was Prince Philip of England. He traced through a maternal line from Queen Victoria. And lo and behold, they didn't match. But they did have, there was a claim that Anna Anderson actually had been an individual called Gertrude Shanskowska. And she had relatives through maternal descent that still were around and were able to test, and their mitochondrial DNA did match Anna Anderson. So she really was this Polish woman who probably did think she was Anna Anderson. She apparently had had some stressful times and some mental problems, and probably did think she was Anastasia. Some cautions about mitochondrial DNA. It changes very little over time. A match may go back dozens of generations to a common ancestor. For that reason, if you just get a stray match and you're not actually testing a question, it's not going to be all that useful. I get matches, full mitochondrial matches all the time, and they're meaningless because I've never heard of these people, nor their ancestry. Somewhere back there, we probably connect, but it's a long way back. The complete matches are the most valuable, the full genome of the mitochondrion. And again, family tree DNA has the largest group of testers. All right, the big one in genetic genealogy is autosomal DNA. That's 90% of the DNA in your cells is autosomal. Autosomes are the non-sex chromosomes, the other 22 pairs. So there's lots of DNA there to match. Couple of issues with autosomal DNA. If the DNA divided nice and evenly during the production of eggs and sperm, it wouldn't be quite as bad. I mean, it's still going to be bad because of recombination at fertilization. But what happens during cell division is the chromosomes line up and will shift segments back and forth between pairs, which results in unique combinations of DNA. So if the red here represents your father's chromosome in you, this process, which is called crossing over, results in unique combinations of DNA in the chromosomes. So what it means is that while theoretically you inherit 50% of DNA from each parent, it varies a little. So this crossing over allows mapping of DNA. And again, the term applied to map units is centimorgans, distance units, which I've already talked about. But theoretically, you get half your DNA from mother and half from father, but it's not exact. It's about 50%. From your grandparents, it should be about 25%, but it starts to vary even more. You may only get 15% of your DNA from one grandparent because of crossing over. Great-grandparents, 12.5%, great-great-grandparents, 6.5%, great-great-great, 3 and 0.125. And then it starts to to get less and less and less and less. So effectively, you can't count on getting any DNA from some of your great-great-great-grandparents. You can generally figure you're going to get at least a little bit from each great-great-grandparent, but again, that may vary. You may have one great-great-grandparent where you get no DNA. But for the most part, it works pretty well back to great-great-grandparents. So that's why if you want to test hypotheses about ancestry using DNA, and you want to see if you trace back to Charlemagne, well, you can forget DNA for doing that. It's not going to work. You would share too little DNA, if any, with Charlemagne. And this is illustrating it graphically. Most of the matches I get notified about, MyHeritage is very good about this, are third to fifth cousins. Look at the purple and blue and the number of segments you're going to share with a third to fifth cousin. Not very many. So I tend to ignore third to fifth cousin matches unless they've got a tree that shows something that interests me, that matches up with my tree. So what do you get from autosomal DNA? When you get results back, you get the amount of match with others in centimorgans. Oftentimes, depending on the company, you'll get the chromosome where the matches occur and the segment location. The higher the number of centimorgans of shared ancestry, the closer the relationship. The general rule of thumb is if you're under 10 centimorgans, that could be due to chance. You may not be related to the person at all. So 10 and up, and the more the better to indicate relationships. So in general, people use this a lot for ethnicity. You've probably seen that on TV shows like Finding Your Roots, or ancestral pathways, and I'll show you examples later. These provide clues or confirm your paper trail. And I always want, if I get a relationship match, if I get a match, I want to use the paper trail to actually confirm how it's a match. It depends on cooperation with other people, with other individuals. And I will tell you, if you only test on ancestry, most of the people on Ancestry, I swear, are doing it just for ethnicity. And they're not going to respond if you try to communicate with them about ancestry. They might respond in 6 months, so they might never respond. If you're really lucky, you find somebody that's really interested and they're going to respond. Here's sample output from, actually this is from Family Tree DNA, and I believe it's my data for me, a match with somebody. Don't ask me who, I don't know. So on chromosome 12, 5 centimorgans, which could be due to chance. I'm not going to put much faith in that. The one on chromosome 10 is 50.75. That, there's something there. That really is a relative. The next three, probably not, maybe. And again, I can't see the last one. So 13.83, yeah, it is probably a relationship there. But if this is my match with another individual, I'm definitely related to this person somehow. And by the way, it gives the exact location on the chromosome where it starts and ends, where the match is. A little bit about xDNA. xDNA is what you inherit from your mother only if you're a man, or if you're a woman from both parents. So what if you look at the DNA on the X chromosome, it actually can give you a lot more information and greater precision if you're looking at a female lineage. So that becomes useful. And I'm not going to talk much more about it. You can read about it, but it restricts the lines that can be useful. And this is a female, and it's showing the lines. This is a fan chart, which shows, the center is the individual, then the parents are the next two layers, the next layer is grandparents, and so forth. And this shows where xDNA examination might be useful for looking at ancestry. So for female, half your dad's ancestry, that is your paternal grandmother's, and then it gets cut by half each generation as you move back for the males. And then on the female side, a lot more useful. But if you look at a man's xDNA, it really becomes useful for maternal ancestry for that man. A lot more useful and focused than for, say, a woman. But even so, for women, it's useful because it's restricted to X only. All right, a little bit about testing. This is usually initially what you're really going to ask a lot of questions about is, where do I test? There's 4 major companies right now: Family Tree DNA, 23andMe, Ancestry DNA, and MyHeritage DNA. You typically pay separately for Y, mitochondrial, and again, those two, you just as well go with Family Tree DNA. Autosomal, separate fee, and I've paid for Autosomal at Family Tree DNA as well, because at one time it was one of the prime companies, still is, but it has a smaller data set. xDNA you derive from Autosomal if you're going to use xDNA. Each company sends a kit with a solution in tubes and a mailer, and some operate on spit, like Ancestry. Some operate on cheek swabs. For Family Tree DNA, I had to do a cheek swab. And you get enough cells in both methods, and the stuff in the tubes is a buffer that maintains the DNA so that it doesn't deteriorate. And you just put it in the mailer, and it's prepaid if you've paid for your test. And once they got your DNA, you don't have to get tested again. You just pay for new tests or additional tests. The company will still have your DNA. One difficulty there, a lot of times elderly relatives have trouble with the spit test. And I believe the ones that do that, you can actually request cheek swabs in some cases. In other cases, you just have to try to get enough spit. And you don't do it, you're not supposed to eat ahead of time for, I don't know, some hours, because your food can have DNA in it from the food. So, and using water to increase the spit can dilute the DNA. So you really need to do spit and cheek swabs and follow the instructions. You have options to allow data sharing with most of the companies, and there's a variety of privacy settings. If you do not like sharing too much, you can set the privacy settings. Read that closely. Do what you prefer to do, keeping in mind that if you set the privacy settings too restrictive, that restricts what you can do later with ancestry uses. Results take a minimum of 6 to 8 weeks. By the way, I've also been tested to try to identify an uncle who died in Japanese POW camp. The Army does its own separate testing. You can't say, oh, I'm at Ancestry or I'm at 23andMe. They do their own testing, and they sent cheek swabs. The data you get will be number of matches for Y. So 37 out of 37 in the case where I matched with the other Eddleman. You'll get matching regions for mitochondrial or the entire mitochondrial genome. And you'll get raw data for all chromosomes for autosomal. And some will give you the number of Cinemorgans when you look at matching with other people, like Ancestry does that. So if we compare companies, the primary purpose of the different companies, 23andMe is very big on medical applications. Most of them are concerned with personal ancestry. Again, autosomal for some, family tree DNA does Y, and mitochondrial. And I think one of the others is getting into that as well. The cheek swab ones are family tree DNA and MyHeritage, and living DNA is a new one. Otherwise, 23andMe and Ancestry do saliva. All of them pretty much store the samples indefinitely, minimum of 25 years for family tree DNA. If you have a GEDCOM file, you can upload with some companies, you can upload to GEDCOM from some companies, but not from 23andMe and not from Living DNA. You can upload raw data from one company to the other in some cases. 23andMe and Ancestry and MyHeritage can be uploaded to family tree DNA, which I've done in the case of most of them. And Ancestry will not allow uploads. MyHeritage does for the other 4, plus an additional one called WeGene. And Living DNA also allows uploads from the other 4 or 2. As far as comparison, this is best for ethnicity, and Ancestry has the biggest, 19 million. 23andMe has 12 million. The smallest of the big ones is family tree DNA, only 1.4 million. And the problem is they're not distributed evenly across the world. Chromosome browsers available for most except Ancestry. And the product launch date, that means how long they've been around. 23andMe and Family Tree are the longest. You'll want to try to use company tools a lot of times to analyze what you get. These assist you with matching information and other information. Most of them are common to all the testing companies. They'll take data from all of them, but each of them has a unique feature. So most of you, if you've done this already, probably have done Ancestry. This is some of my output from Ancestry. And it told me my ethnicity, which by golly, I could have predicted that already. It's if you've done genealogy for a while, you rarely get surprises with ethnicity. So I knew I was British Isles and Germany, pretty much. Now, these change over time. The reason is that they keep enlarging their database and improving their reference collection of people in different parts of the world. Yes, and I have 1,000 plus fourth cousins or closer, most of which doesn't tell me a whole lot. Ethnicity estimates are available for most of the companies. Most people test for this reason. It is also the least useful for actual genealogical use because they're constantly refining their ethnicity estimates. It's funny too, a lot of people say, well, this is worthless, it keeps changing. That's called science. Science is constantly refining things, and so it is going to change. I've had Scandinavia pop up briefly, and then the next time they upgraded it, it disappeared. And it's just because of their reference collection, reference populations. What about Native American ancestry? Well, a little bit about that. There are very specific markers that point toward Native American ancestry, but a couple of cautions. One is to remember, you get past great-great-grandparents, you may have no DNA from an ancestor. You may have Native American ancestry with no Native American markers in that case. If you are saying your great-grandmother was Native American and you come back with no markers, I would really start to question that, because there should be some markers there that would point toward that. It goes down to accepting family lore over actual data. One of the things Ancestry does too is they plot out these ethnicity estimates, and then they also look at communities when you get to the US. And I knew this already too, but if you're just getting into this and do DNA, and it says Carolina Piedmont settlers is one community, wow, well, that's useful. That narrows down my search area potentially. And then Southeast Missouri settlers, even more. It also has a secondary area of Tennessee and southern states, which, yes, that matches right up with what I knew from the paper trail already. Then also Ancestry will give you your DNA matches. My sister has tested, and by golly, she's my sister. A couple of first cousins have tested, and yep, they come out as first cousins. And then I have a second cousin that's tested, and yeah, that matched up too, except she's actually a first cousin once removed, but similar amount of Cinemorgan match. Then they do through lines, which you can actually look at who matches up with you. And I'm going to look at my grandfather, C.C. Eddleman. I have 3 DNA matches that are pretty hefty ones. And it ended up, I knew the people already anyway. My aunt Vera has a granddaughter that tested, and yep, came out exactly the way it should have. I know who it is already though. And then one of my other aunts had a great granddaughter who tested, and pretty much lined up with what it should have been. Here's another one on my mother's side of the family. And this one, you'll notice that one, third cousin, 10 Cinemorgans. Well, I know from the paper trail that this probably is real, but 10 Cinemorgans is a little marginal to be worried about. And it's a third cousin level, but I know it's real in this case. And then my first cousin came out on this one too. Third-party tools, these are things that don't come with the companies. There are methods of analysis and interpretation from sites other than the ones maintained by the testing companies. I'm not going to talk about DNA Painter, but that's one that's used quite a bit. One that is really useful and can give you a kind of a clue is the Shared Cinemorgan project. People sent in known relationships and the number of Cinemorgans that matched up between individuals and the relationship. And then this is all compiled together and gives you a range of Cinemorgans with different relationships. So we'll just look at a couple. Great-grandparent, the average is 887 Cinemorgans, but it ranges from 485 to 1,486. You get overlap. And because you get overlap, the Shared Cinemorgan project also has this feature. You can enter the total shared Cinemorgans for a match, and it will tell you the probability of different types of relationships. In this case, 124, 47% chance it's a half second cousin, a second cousin once removed, a half first cousin twice removed, or a first cousin 3 times removed. That's getting down there, but it is real because it's 124, which is pretty large in the whole realm of matches. Then you go down and there's less likelihood for other potential relationships. There is another service called GEDmatch, which you can upload your raw data to GEDmatch and find people that match with you, and actually in this case, where they match, what segments, what chromosomes, what locations on the chromosome. So it allows comparison with a larger population of testers. So initially I uploaded from Family Tree DNA, which has a relatively small group of testers, which allowed me to plug into a much larger group of testers. However, this is the one that got into trouble because of security issues. I won't really say it's security issues per se, but what they found some serial killers by plugging into GEDmatch with some DNA samples from murder victims from murder sites and looking for matches. Well, now you can opt out of that if you so desire. I don't care, if somebody wants to use my DNA to find a serial killer, I am fine with that. But some people don't like that idea, in which case opt out of it. So it's been partly dealt with. So to upload, you got to fill out a form, check the email for confirmation, your email, and it'll give you a confirmation email with a code, which you have to enter. And then you can upload results to GEDmatch. And you can do this, I believe, from all of them, but Ancestry and the smaller ones. And when I, do just a general fishing expedition, I do one-to-many matches, that option for raw DNA raw data. And it'll ask for your kit number, which is how you're identified in GEDmatch, as well as in the company where you tested. And you can set the minimum Cinemorgans. 7 is like really minimum. Anything below that is likely not real. And then it will spit out kit numbers and the total Cinemorgans matched. If you want to go farther in learning about DNA in genealogy, the best single book, and it's not all that thick, and it's very readable, is Blaine Bettinger's book, Guide to DNA Testing and Genetic Genealogy. If you get a hold of a copy, make sure it's the 2019 edition. There are earlier editions out there. The 2019 is updated considerably. Then the handout has some additional information. And this book is also available from the National Genealogical Society, Genetic Genealogy in Practice. It has exercises at the end of each chapter where you can kind of apply what you should have learned in the chapter or test yourself. And that's available from NGS. I want to make a point with some of these. The Lost Family, Finding Family, The Foundling. These are all popular books. Most of them pretty readable, pretty interesting. But I will caution you, if you're stepping into genetic genealogy, don't do it if you don't want a surprise. There is the slim possibility that you will find out such things as your father really wasn't your father. There are secrets in the family that nobody told you about, and you'll uncover them with DNA. And if you feel that will be a problem, don't do it. Some people have had to go into counseling over finding these things out. I'm talking to another genealogist that had a client who actually was threatening suicide over finding out her father was not really her biological father. So expect surprises, although they're a slim probability, they can occur. And it'll be devastating to some people. So we should have answered most of these questions. What's DNA? Can it tell me if I have Cherokee ancestry? Another point about Native American ancestry, it cannot tell you tribes. There was considerable intermarriage among tribes. The tribes that were present when Europeans settled in a given area may not have been the original tribes anyway. So it can't tell you that. I had somebody approach me once wanting some work done. They said, well, I tested with this company in Canada, and they said I was 1/10 or 10% native ancestry, 1% was Cherokee, and 9% was Cree. And I said, you better check up on that company. That's not ethical, because it can't identify tribal affiliations. And Cherokee were in the southeastern US, Cree were in northern Canada. I don't think so, especially in the time frame this guy was suggesting. Can it substitute for traditional research? I think I said numerous times, always go to the paper trail as well. It's just one more piece of information. It's not the grand do-all and end-all. You can use it to assist with constructing a family tree, but it's not going to spit it out for you. Nope, you don't need to exhume your ancestor because the DNA is going to be in somebody else. It would have been nice to have tested that ancestor if you want to test ancestors farther back, but you don't have to do it. No blood sample needed. It's all done with cheek cells and saliva. The results are as private as you want to make them. And the tests you have done depend on what you're trying to prove, really. A few final thoughts. It's best not to test as a fishing expedition, which a lot of people do. But these can be interesting, but it's expensive if you're just fishing for something. It's not the magic answer to all your brick walls. It helps. It's one more piece of information. But, you know, I can find even better stuff if I find the right land deed. Genealogy still depends on doing the best research and documents. So it's just one more thing that you can use. You can use it to confirm suspected ancestors primarily. That's what its use is for. Read and ask questions to understand the tests. The companies will answer your questions. They have websites where you can ask those questions. So utilize it. You paid for it. Get tests done while relatives are still with us. My last uncle passed away 3 weeks ago. Several of his kids got tested, so it's probably not essential that he would have been tested, but again, it might have clarified one more generation back. As with any source, you get better at it with practice and reading about it and learning about it. So I hope you got a basic introduction to the use of DNA. You can do whole courses on this that last a week and then do an advanced course that lasts a week. There's a lot here. There's a lot more that I didn't talk about.
Thank you so much for another great program.